Okay, welcome everyone. We're going to get started in just a minute. Let everyone come in from the waiting room. Okay, thanks for joining us this afternoon. Welcome to Let's Chat About, the free monthly webinar series hosted by Hope and Focus. We've developed the series with those living with LCA and IRDs in mind, but it is open to anyone who is interested in what's happening in our communities. My name is Courtney Coates, and I'm the Director of Outreach and Development for Hope and Focus. Before we get started, I just want to take a moment to thank our sponsors for this series. They are AGTC, Dominion Energy, Editas Medicine, Janssen, Mira GTX, and Spark Therapeutics. We could not provide programs such as these without their support. A few short housekeeping items. The session will last about 45 minutes. Your microphones have been muted and your cameras are off as this is a webinar format. You can submit questions through the Q&A function or the chat function, and as time allows, we will take any questions that come in live. We are recording this session, and after the webinar, we will post the recording to our YouTube channel and we will send out the link. It is my pleasure today to welcome Dr. Daniel Chung, Chief Medical Officer for SparingVision. SparingVision is an ocular genomic medicine company focusing on gene-agnostic gene therapy and CRISPR gene editing approaches to combat blinding diseases. Prior to his recruitment to SparingVision, Dr. Chung was the ophthalmology therapeutic leader for Spark Therapeutics, where he led the medical affair group and contributed to areas of clinical development and operations, marketing, commercial, patient advocacy, preclinical research, and development and business development. Dr. Chung was intimately involved with the development of Luxturna, the first gene therapy approved by the FDA and EMA for use in a blinding genetic disease. Prior to joining Spark Therapeutics at the Shea Eye Institute, University of Pennsylvania, working in retinal gene therapy, also serving as a sub-PI on the RPE65 gene therapy clinical trial at the Children's Hospital of Philadelphia, and made major contribution to the development of the phase 3 MLMT novel endpoint, was PI of the MLMT study and the RPE65 natural history study. He completed his ophthalmology residency at Summa Health Systems in Akron, Ohio, and then completed fellowships in pediatric ophthalmology and ocular genetics research at the Cole Eye Institute at the Cleveland Clinic, and in retinal gene therapy at the National Eye Institute / NIH in Bethesda, Maryland. Please join me in welcoming Dr. Chung.
Well, thanks, Grace, for the introduction. I know that was a mouthful. It's great to be with you, and thank you again for the invitation.
Thank you, Dan. We appreciate you being here. So today's conversation is about gene-independent therapies for IRDs. Can you give us a basic overview of what a gene-independent or gene-agnostic therapy is?
Sure. So a lot of the gene therapies right now, especially in the ocular retinal segment, really target a specific gene that patients with variants or misspellings in that gene are eligible to be part of those gene therapy trials. And they really are trying to basically restore the function that is lost in those patients that have variants in that specific gene. For example, RP65 retinal gene therapy is very specific for patients who have misspellings in the RP65 gene. Right now, there are over 300 different genes that cause inherited retinal diseases. And so there are a lot of great programs out there by a lot of great companies and academic institutions that are investigating a lot of these genes. But obviously, trying to develop all 300 of these genes will take quite a bit of time, as well as a bit of resource to do that. So what we are trying to do at SparingVision is really look at some of the ways that we could do gene-independent approaches, which means we're not necessarily correcting the genetic defect, but using other genetic parameters where we can help slow down the degeneration of disease in a gene independent portion or way. And how does that actually work? So for us, we're really looking at preserving cone function. So if you know in the retina, there are these light sensing cells called photoreceptors. So there are two basic types. And one are rods, which are in the periphery of the retina, and they're really more for our nighttime vision and side vision or dim light side vision. And then cone photoreceptors are more in the middle of the retina, and that's what you're actually using on a day-to-day basis for your best acuity, color vision, and daytime vision. And so what we're looking at, and what our founders, Professor Jose Sahel and Thierry Livillard, originally asked the question, most of these retinal genes cause defects in those rod photoreceptors. So we understand why patients have loss of their side vision, of dim light vision, and things of that nature. But then why do majority of these patients, we call them rod cone dystrophies, a type of that would be retinitis pigmentosa. But the question was, why do these patients then also have defects with their central cones as well? Because the genetic defect doesn't really go to the metabolism or the function of those cone cells. And so they basically look to see, is there some connection between these two cells? And what they were able to identify is a factor that they called rod-derived cone viability factor. And basically, this is a survival factor or a factor that occurs naturally in everyone's eyes, where these rod cells that are on the periphery actually secrete a protein that actually attaches to cone cells, and through this long mechanism of metabolic activity, helps to protect the cone cells and keeps them functioning and their structure moving forward. So that's what happens normally. But in diseases where you actually have rod cell death, you don't have that factor being produced anymore. And that's why it's gene independent, because it really doesn't matter why your rods are dying. It simply matters that they're not there to produce that factor, and that factor is necessary to keep your cones functioning. So by using gene therapy, we're putting that factor back into the eye of rod cone dystrophy patients. And hopefully we are able to slow down then the degeneration of your cones. And your cones are, again, for your central vision, daytime vision. And we hope to be able to extend that a significant period of time. As you know, most retinitis pigmentosa types have a relatively slow degeneration. So if we're able to slow that down by 40 or 50%, you would have significantly longer usable vision. We're not really doing anything for the rods at that point because they've degenerated. We're not restoring them, but at least we can hope to preserve the function of the cones. Now, that's our first program. We call that SPVN20 with the rod-derived cone viability factor. And there's another factor because the gene actually produces two different forms. One is the secreted form, or the form I just talked about. And then there's another form which acts as a powerful antioxidant. And we've all heard of antioxidants. You got to eat your blueberries and your cranberries and all these things. And that's something that works within the retina as well, because the retina uses more oxygen than any other organ in the body by gram weight. And because of that, there's a lot of reactive oxygen species that are produced. And so you really have to dampen those. And that's why this other form of RDCVF, we call it the long form, works by being an antioxidant and reducing that. So that, coupled with the other property of the secreted form, the two act together to help cone survival. Now, then we have another program which will use a gene called GIRK, which stands for a G protein inwardly rectifying potassium channel gene. And this, we believe, will actually restore function in cone cells that have lost the ability to function, but their cell body still remains. So that's what we see a lot of times in degeneration, in retinal degeneration, is we see that the function is lost and certain parts of structure might be lost, but the actual main cell body stays intact. And what we use with the GIRK, and this was developed by our chief scientific officer, Dr. Denise Dalcara, has been able to show that you can revive those dormant, we call them dormant cone cells, and restore that cone vision. Again, doesn't matter why they degenerated. So it's not gene dependent because we're not correcting the gene that caused the degeneration, but we're adding another factor in that. So those are the reasons why these are what we call gene independent approaches. That was a very long answer. So
Thanks, Dan. We appreciate it, though. It's a very interesting sector to be in. So hearing that those are the three things in the pipeline, how is this a good option for those with potentially rare gene mutations or those that have low vision but still some cone function?
yeah, it's a great way. We hope that we'll be in human trials very soon. And the idea is that if we can restore function with the GIRK product, we call that SPVN 20, or with the RDCVF and trying to slow down the disease in patients that are a little less affected, that still have a fair amount of cones, we want to preserve that as long as we can. The reality is that when it comes to a lot of these really rare diseases, unfortunately, a lot of the really ultra rare diseases may have some difficulty getting traction with development just because there's not a lot of patients that have those genetic defects. But this is a way that even those patients that are very rare, that have rod cone dystrophies, these patients could also, or these individuals could also be potentially treated with these gene independent approaches. Because again, it's not about why you have rod cone dystrophy, because we're not technically treating the genetic defect, but we're trying to preserve vision of what you have left.
Great. And I know we recently just came from the Visions conference in Orlando. How does this differ a little bit from things like optogenetics or stem cells as well?
Yeah, so optogenetics, a lot of times optogenetics deals with the idea that, at least the initial thought is that optogenetics is when individuals have no photoreceptors left, and we call that the outer retina, and those light sensing cells are no longer present. And so what a lot of very good groups have done is they've looked into using different ways of actually making inner retinal cells, and a lot of times these are ganglion cells or bipolar cells, more light sensitive. So they're basically trying to make them act like somewhat like photoreceptors or light sensing cells, and being able to restore some of that light sensitivity. I know that there are other folks that are actually looking at cones as well, to do some type of optogenetics. So optogenetics is another gene independent approach. We do think that some of the things that we do will be for patients who aren't quite as affected as some of the candidates that are being going through clinical development trials. So we hope to be able to address disease a little earlier in the progression of things. And your last question, the second part of that question was?
And stem cells.
Oh, and stem cells. So stem cells really, again, are for a lot of end stage disease where you don't really have those light sensing cells left. And the idea is you want to put in stem cells to hopefully regrow those and to be able to have some light sensitivity then. So those are very much more end stage when you actually have lost all your cells. Whereas for us, for the 06 program, we want to just save whatever cones that you have remaining. And then for the 20 product is you still have those cone bodies left, even though they're not functioning. And so those are the things we want to try and revive.
Great. So individuals in our community can kind of look at what level of vision that they currently have and try to find the best fitting therapy for them.
Absolutely. There are a lot of different entry points in a lot of these different gene therapy trials. A lot of times the trials that are doing specific single gene therapies, they might want to be a little earlier in disease, where a lot of them are really trying to regain some light sensitivity and so rod function. But every gene therapy is a little different. And so it's just a matter of really investigating those to see what really fits your disease and your progression in that. So.
Great. And it's great to hear that, you're looking towards some potential clinical trials in the next couple of years. So it's been great to hear that there's more treatment options becoming available in the next few years for our community. I do want to let all those that are attending know that feel free to drop your questions in the Q&A or the chat, so that Dan and I can include those in our conversation today. I've found it very interesting. We've, been talking a lot about gene independent therapy recently, but I just want to talk about the importance of still finding out your genetic diagnosis. And if you could speak a little bit to that.
Sure. I think there's been great movement within the community in the last 5 to 10 years when it comes to gene therapy. I mean, when it comes to knowing your gene. There are a number of free programs out there that you can enlist to find your gene and get genetic testing. I think genetic testing is very important because that is one of the ways that you really can get a definitive diagnosis as to what is causing the retinal degeneration that you're experiencing. And this can help your physician as well as yourself to understand a little bit more about the disease, potentially progression rates, and things of that nature. And also to be connected with the community. There are a lot of great patient organizations like yours here that really are somewhat not totally specific, but they might be more for RP or LCA or choroideremia or something like that. And then when it comes to actual gene therapy trials, some of them that are gene specific will need that genetic diagnosis. And so genetic testing is not foolproof by any means. It doesn't always give an answer. But you just need to make sure that it's done by a good reputable lab. And then it's always good to follow up with a genetic counselor to really help you interpret some of those results, as well as talking with your physician. So even though we might be gene independent, I think that the idea is we still want to encourage people to get their genetic diagnosis.
Great, thanks. I appreciate that, Dan. No questions in the chat yet, but hopefully we will get some in shortly. In the meantime, is there anything else in SparingVision's horizon that we can look forward to?
Sure. The one thing is we're not technically just a gene therapy company. We totally think of ourselves as a genomic medicine company, and that's really using all the genetic tools that are in the toolbox to help combat these inherited retinal diseases or just ocular diseases in general. And I think we also have a partnership with Intellia Therapeutics where we are hoping to address some of the inherited retinal diseases that may not be the best choice in doing gene therapy because either the gene's too big or there's some other complicating factor that maybe we can do gene editing. And we've all heard of CRISPR. Very simply, it's like a molecular scissors where we can cut in certain things out that will help that would help decrease some of the disease outcomes in some of these inherited retinal diseases. I will say we've gone from one end where we're gene independent, and then we've gone all the way to the other side where CRISPR is more, not only is it gene dependent, but it's also dependent many times on the misspelling within that gene. And each gene has a lot of different misspellings that can cause disease. And so this is a little more targeted. So it won't be necessarily for everybody, but it'll be for those individuals that have certain misspellings. Now, the way we do this, a lot of times we can cover the whole gene by having slightly different strategies for groups of those misspellings. But we're also looking at that as well. And so those are some of the things that we have on the horizon. And we're starting to do some work on that in our preclinical stages as well. So
you're really at the two ends of the spectrum.
We really are. And it's really just the idea that we just want to do as many different strategies as we can to really try and get more options for patients. I think obviously it would be great to be in a world where it's not a matter of having one therapeutic option, but to have several therapeutic options that really is tailored toward where you are in your disease progression and what fits best for you and the inherited retinal disease that you have.
Yeah, one of the questions that I was asked recently is, can individuals potentially get two different types of therapies or treatments depending on their disease stage?
Sorry about that. Let me just get out of this call here. Apologize for that.
No.
One of the things about gene therapy is that we're still in very much the early stages, and what we have seen is that the example of Luxturna is that we have been able to see that injecting the same gene therapy product in two eyes of the same patient appears to be relatively safe. There's been a good safety profile on that through clinical trials and now with commercialization. The idea too is that very similar vectors can also be injected in the two separate eyes as well, with a relatively good safety profile in the sense of, for RP65, there were slightly different vectors from some different companies. In other words, they were able to receive their first product from that other company through their clinical development program. But I believe that some patients have now received Luxturna in the contralateral eye with no other safety issues that I know of. Now I'm not with Spark Therapeutics anymore. So the question that you asked, though, is can you receive two different gene therapies within the same eye? And I think that question is yet to be answered. I think there might have been some studies looking at injecting the same eye with the same gene therapy product. And then, and I think those are still preliminary. Those are mainly in animal models. But what you're talking about is, can I receive maybe a gene therapy for RP65 Luxturna, and then could you receive ours because it's slightly different? And that question has not been answered yet. That still has to be some safety studies that have to be done both in animals, and ultimately, if that proves safe, is to do a clinical trial possibly to see what that would be like. But that is a question that many people ask because obviously there are a lot of gene therapy programs out there, and they want to know if something else came down the road, if they could do that. And I would say the question is still unanswered.
Yeah, it is. Now that there's a lot out there, it's a little confusing as to, do you wait, do you move forward? But it's exciting to have these choices. We did have a couple of questions come in, so I'm just going to take them in order. Lori said, any chance on letting us know what genes they will be working on in the near future? So I assume this is more about your gene editing program.
So we have not disclosed those targets as of yet, and we're doing some preliminary work on that. Even in our gene agnostic or gene independent approach for SPVN06, the RDCVF program, we need to start with some specific genes to start with, just because you have to do some preclinical work on that with animal models and things of that nature. But then we'll be adding more and more genes, we hope, and to get to a place where we can be truly gene independent.
Great. And the second question is, are there any precedents for CRISPR being used successfully to treat a genetic disorder outside the visual system? If nothing is currently available yet, what are some of the approaches that are in development?
Yeah, so Intellia Therapeutics has actually had some success, had significant success as one of the first, I believe, to show that they've been able to alter the human genetic sequence using CRISPR in a systemic disease. I know that gene editing has been used with a company called Editas in a form of Leber's congenital amaurosis, and that trial is ongoing as well. There are a number of other companies using CRISPR technologies, both in the eye and outside the eye. So it is definitely getting quite a bit of traction. And if you go on to clinicaltrials.gov or look up Intellia Therapeutics or Editas, I'm sure you'll be able to find some more information on those programs.
Yeah, that's great. Thanks, Dan. And I'll just remind everyone, keep the questions coming. We will definitely try to get those answered today. Okay, just gonna give everyone a minute to type those in. But appreciate you being here today, Dan. I also appreciated listening to some of your talks at the Visions conference this past weekend. There was one kind of analogy that you shared during the LCA research updates about early, mid, and late stage treatment options and how those might apply across those. And it was really helpful to hear that, some of these treatment options aren't just for those who have some vision, that there are some options beyond that, and finding what fits is really important for where everyone is at in their progression.
Yeah, that's really is the idea that there is something almost for everybody when it comes to the progression of disease. So early on, when you still have some rod cells left and good cone function, or even with decreased cone function, a lot of times early on is really a lot of your gene specific approaches are targeting a specific gene. And then on the other spectrum is those patients that really don't have a lot of visual function left, and they look to the stem cells or the optogenetics or actually the retinal prosthetic as well. Those are all options that are on the table that are currently being researched, some in clinical development. And then there are the ones that we talk about cone preservation. So you still have to have some cones left. We're trying to slow the degeneration of that. And then, of course, our GIRK program, our SPN20, is also about restoring vision in somewhat later stage disease. So everybody's trying a different modality in the sense of trying to really address the idea that the natural history of disease has many different stages, and that we hope to be able to at one point hopefully have strategies that can address a lot of those different times in the progression of disease. I did want to mention one thing, because I did mention clinicaltrials.gov. I think it's very important to say that you have to be a little careful and somewhat inquisitive on clinicaltrials.gov. I think there's a way that you can sort out the trials on that website that are actually FDA reviewed and approved. And so that's simply to say that not everything on clinicaltrials.gov has gone through FDA approval or regulatory approval. And so you just have to be a little wary of that. I do think there's a button you can hit to filter out any of those that are not regulatory approved. So.
Great, thank you. Yeah, that website can be a little bit overwhelming sometimes at first. So using those filter options are very important. Okay. Well, if we don't have any more questions from the audience, I'll wrap us a little bit early today, but I've really appreciated everything that you've shared with us today, Dan, and we will have this recording available for those who would like to view it at a later time. I just want to remind everyone that our next Let's Chat About will take place on June 30th at 1 p.m. with Kathleen Hayes of SmithSolve. And that's going to be on the results of our LCA community survey that we've conducted the past couple months. It's sure to be a great conversation. We have some exciting results to share. So we hope you can join us for that and sign up on our website. Okay. Thank you so much, Dan. We really appreciate your time today. I know you have a very busy schedule, so we're very thankful.
Pleasure to be with you again. Thanks for the invitation, and I'm sure we'll see you soon at another event.
Thank you. Goodbye, everyone.